Hello champions, how are you all? I hope that you are doing wonderful and I welcome you all to PU2 Brahmastra Series. Today is day 24 and today we are going to deal with this chapter Biotechnology, Principles and Processes. I saw a few of you telling me, ma'am, can you please complete Biotechnology? I hope you're talking about this chapter, and if you're talking about Biotechnology and its application, it will be really completed soon. So do not worry about any bit of it. Okay. Now, one thing that I want to tell you truly is that I do not see active participation from your end, which is truly dissatisfying and saddening, because we are here standing and talking, okay, basically continuously for hours just for you guys, so that you get the best thing, the best deliverable in the limited amount of time, because we also know for 3 hours also we can keep on talking, but our energy also goes low and it won't be really entertaining for you guys also to watch it for 3 long hours. So please respect these efforts that we are putting. We definitely respect all your efforts that you are putting, okay. The comments, the likes, the shares, everything means a lot to us. Please continue to do so. Please watch the videos. The exams are coming. Let us know that the efforts that we are putting has some value in itself. Now, if you're thinking that it's okay, ma'am, from today onwards we are going to change, we are going to watch the videos, we are going to make 100% utilization of it, then please comment down that we are in. Okay, so I would be seeing that comments, checking who all commented. Now, next thing, how many questions does this particular chapter, or basically how many marks does this particular chapter hold? It holds 7 marks, my dear children, and according to the 3 blueprints that is given by the government, okay, in 3 of them they have mentioned that there will be 2 1-marker questions, okay, and there will be 1 5-marker question. So you do not expect questions from 2-marker and 3-marker category. So it will be just 5-marker, or else it could be MCQ questions, or basically fill in the blanks question. None apart from that. Okay, so you have to make sure that your concepts are really good, okay, then only you would be able to answer the 5-marker questions. I would be showing you certain repeated questions which have come so far, so you can prepare accordingly. I have taken questions from the model question papers as well. None of the questions I have left, so don't please worry about anything, okay, everything is going to get sorted. Chalo. And one more thing, we have launched the PU2 Success Blueprint Test Series. So if you are someone who is interested in it, definitely there's a link given below in the description. Click on that. It's for $5.99. Okay, it's a test series that is curated by lots of people, and there are 12 mock tests. And after that, we would be providing you video solutions as well as doubt clarification classes. So do not think twice. Register. Okay, make use of it. The first test series would be launched on 15th of December 2025. Cool. Chalo. How many questions are we planning to discuss today? 14 1-marker question, and there will be 7 5-marker questions. Cool? Chalo. The first question says, the steps of recombinant DNA technology are given below. What do you mean by recombinant DNA technology? Basically, suppose I have my DNA. In my DNA, I have a gene, and this gene is responsible for coding some protein. This protein is really, really important for me, but I cannot rely on my body for it to be produced, because it takes time. In our case, replication and everything takes time, and obviously I cannot use that product, okay, because I cannot probably isolate it very easily. So I should be putting it in some other organism whose replication rate is kind of fast, okay, and we can easily isolate our gene of interest from it. Sorry, our protein, or basically our product, okay, our foreign product from it easily. So this is the DNA of a bacteria. And what did I do? I took my gene and I put it over here. Now this is a recombinant DNA. There is a recombination of things, okay. Recombination of things have happened. I have cut their DNA over here and I put mine over here, such that this particular bacteria, they replicate really fast. So they replicate really, really fast, and I get more and more copies of my gene of interest. This is my gene of interest, and if I get more and more copies of my gene of interest, basically I am doubling the amount of protein that it is going to produce as well. This is known as recombinant DNA technology. Now listen to me very carefully. In this, what would be the first step? The first step would be isolation of the DNA. The first step would be isolation of DNA, because, okay, DNA might be combined with RNA, protein, lot of things. So I just want DNA, nothing else. So first step should be isolation of the DNA. Okay. Next thing, after I isolated it, I have to cut this particular DNA. I have to cut this particular DNA over here, and then I have to insert it over there, right? So cutting of DNA at specific location becomes the second thing. Now, after that, what? After I've cut, then what, children? After I've cut, I have to make sure basically I am having multiple copies of this particular thing. Before inserting, I have to make sure I have multiple copies so that I can put these multiple copies in different bacteria. I can put it in different bacteria, right? So basically, I should be amplifying the gene of interest. The next step should be amplification of the gene of interest using something that is known as PCR, polymerase chain reaction. Okay, after I have amplified, now I would be putting my gene of interest into some other organism's genome. So suppose this is the bacteria, this is the plasmid which is there. Plasmid is an extra chromosomal DNA, extra thing. They also have their DNA. Apart from that, they have some free living DNA in there. Okay, not free living, basically they have some extra DNA in them, extra chromosomal DNA which is just there. It does not code for anything. So even if I make some changes over here, does not matter for the bacteria. There won't be any changes that would be occurring in the bacteria. So the next step would be what? Insertion of recombinant DNA into the host organism. So this is the host organism where I am putting my content. So this is fourth option. Now after that, after I have inserted, they will do the process of replication, transcription, translation, everything. I will get my product. Obtaining the product, this will be what? The fifth step. After I have obtained it, I have to make sure that it's clean. Okay, there might be again lot of things along with the product that I am looking forward for. So you have to make sure that it undergoes a processing, and that processing is known as downstream processing, my dear children. So this becomes the sixth point. Okay, so this is the first point for this one. So let's see, in all of them, that one is the first point. Let's see the second point. Second one should be third one. 1, 2, 3. Over here only I see the second point as third. So I cancel this, I cancel this, I cancel this. But still, let me see the next thing. After third, it should be second. Yes. After second, it should be 1. Yes. After 1, it should be fourth. Yes. It is fourth, and then it should be fifth. So this becomes your correct answer. Okay, this is the model question paper question. Going ahead with the next question. Choose the correct sequence of polymerase chain reaction steps from the following. So polymerase chain reaction is involved in what? Basically, I want to amplify the amount of DNA. So basically, for that, suppose I have a DNA, I have to amplify it. I have to open it, right? I have to open this DNA strand. This is known as denaturation, my dear children. This is known as denaturation, which is done at high temperature, very high temperature, almost 95 degrees Celsius, almost 95 degrees Celsius, where the hydrogen bonds and everything is broken down. After things are getting broken down, I have to make copy, right? So for making copy, I need DNA polymerase. For making copy, I need DNA polymerase, because DNA polymerase would be making a new strand. But the problem with DNA polymerase is that it cannot just start by itself. It needs certain primers, certain nucleotides already fixed over here. So these are known as what, children? These are known as primers, and this step is known as annealing. This step is known as annealing, which takes place almost around 50 degrees Celsius. Almost around 50 degrees Celsius, it can go beyond that as well. Okay, now the DNA polymerase basically can extend the primers. DNA polymerase will be extending the primers in 5' to 3' direction. Okay, so this will be 3', it will be 5', this will be 5'. This step is known as basically what? Elongation. Elongation again requires almost 74 degrees Celsius and stuff like that. And this is done by DNA polymerase, but in them there's a specific type of DNA polymerase. You call it as Taq polymerase. You call it as Taq polymerase because you obtain this DNA polymerase from an organism that is known as Thermus aquaticus. Okay, you obtain this from Thermus aquaticus, which is a bacteria that stays in very high temperature. So obviously in all this thing, you might have seen the temperature is kind of high. So your enzyme should be able to withstand it, otherwise it won't be able to do it, right? So this particular enzyme is perfect for that. It is able to withstand everything. Cool. So this becomes the thing. So denaturation, then annealing, then elongation. Denaturation, annealing, and then extension or basically elongation is the same thing. So this becomes the answer. Cool. Next, a piece of alien DNA cannot multiply itself in the progeny cells of the organism due to. So basically, if I have taken the DNA out from this, a gene, and just I am putting this gene in a host organism just like this, will it be able to replicate itself? Will it be able to multiply itself? No, children. If I want it to multiply itself, I have to put it inside a plasmid or anything which basically has one very important part, which is known as ORI, that is origin of replication. If origin of replication site is not there, the DNA polymerase would not be coming and attaching and replicating itself. So that is a problem. So a piece of alien DNA cannot multiply inside the progeny cells. Okay, it is not able to multiply. Why? Because it does not probably have an ORI site. There is a lack of ORI site. Cool? This is a model question. Now, hint 2 cuts DNA molecule by recognizing a specific recognition sequence of what? So this was the first restriction enzyme that was being found. So basically, hint 2 is basically a restriction enzyme. It has to cut the DNA sequence. It has to cut a DNA sequence. But how many base pairs should be there? Almost there should be 6 base pairs. 1, 2, 3, 4, 5, 6. There should be 6 base pairs, okay, in which this is a recognition sequence. What do you call? The sequence where it will come and cut. It is fixed. It's not given in your NCERT, so I don't even want you to remember it, okay. But this particular sequence would be fixed. For example, the thing that is given in your NCERT is EcoR1. EcoR1 is again an enzyme, okay, and the sequence of it is GAATTC. This is the sequence. So this sequence only it will be cutting. Nowhere else it will go and make the cut. It will recognize the sequence and make a cut over here. Similarly, hint 2 also has this, but how many base pairs are there? 6 base pairs are there, my dear children. In your NCERT it is given 6 base pairs, but they didn't mention about the bases there. Cool? Chalo. Next. Given below is a portion of DNA strand giving the base sequence on the opposite strand. What is so special shown in it? So here 5-GAATTC is given and then 3- is given. Here 5-GAATTC is given and 3- is given. What do you call this type of sequence which is read in the same way from both the sides? The sequence which is read in the same way from both the sides. For example, you have the word Malayalam. Any Malayalis up over here? Well, I am a Malayali. Okay, so over here, it can be read Malayalam in this way also, the same thing appears. Similarly, here also GAATTC from 5- to 3- end. Similarly, GAATTC from 5- to 3- end. These sequences are known as palindromic sequences. This is not a start codon, children. This is not deletion mutation. You cannot say replication is completed because anyway, whatever the sequence is, AT, GC, and then GCAT. Okay. Yes? Yes, right? Whatever way, in this way, replication is anyway completed. You cannot say there's anything special about it. Here, over here, the thing that is special is that it is read in the same way in both the sides. So this becomes your answer. That is, it is palindromic sequence. Cool? Ayyo. Yes. Next question. Plant cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in a method known as. So why it is done? So basically plant cells, if you have seen them, okay, plant cells does not just have a membrane. It does not just have a membrane, that is plasma membrane. Apart from it, it also has something which is known as cell wall, my dear children. It also has cell wall, and to pass through cell wall it is kind of very difficult. So what is done is that there is something that is known as gene gun method. Gene gun method, where what happens, the microparticles of gold or tungsten are being taken, okay, the microparticles are being taken and it is coated with the DNA of interest. It is coated with the DNA of interest, and then just like a gun, it is being put over here into the nucleus of the plant cell. Now this method is known as gene gun. Why gun is used? Because to pierce through the cell wall it is difficult, which is why a gun is being used. What do you call this? Gene gun method. Now over here you do not see gene gun, so what do you do, ma'am? So basically biolistic is the other term for gene gun method. So please make sure you know about that. This is given in your NCERT, but people tend to forget it. Okay, so for plant cell you have gene gun method. Why? Know the logic behind it. It's because they have a cell wall which is extra thick. Okay, now micro injection is there where you're directly going to put the content into the nucleus. Now this is done in the case of animals. Okay, heat shock is basically done in the case of bacteria and stuff like that. Vector mediated, again it can be done in animals, plants, anything like that. Okay. Chalo. The commonly used vector for cloning genes in animals is, the commonly used vector for cloning genes in animals is, vector means the one who can probably carry the gene of interest. Okay, the one who can probably carry the gene of interest. Now, basically, suppose I'm just giving an example. There is a bacteria that affects a plant. With the help of a unique DNA that it has. Now what I am doing, anyway it has the machinery to go and affect the plant. What do? I do certain changes over here. I remove the piece of DNA that could cause the problem. Instead of that, I put my gene of interest over here. Anyways, it is going to affect. When it is going to infect, it is going to put my gene of interest over here. So am I not passing my content over here, the gene of interest that I wanted to, right? So this is what this guy is going to act like a vector. So you are asked about vectors, not in the case of plants, but in the case of animals. So in animals, the commonest vector that you use are viruses. Okay, viruses, because they know really well, they adapt to our body really well. Okay, you know coronavirus, that how difficult was it to eradicate it completely. It is not completely eradicated, but still. So you can use retroviruses, which are a group of viruses, okay, as a vector, but you have to make sure that they are disarmed. Disarmed means basically I am removing the DNA, or basically I am removing the genetic material which is going to cause the trouble. Similarly over here, I have to remove the content that is going to cause the trouble. So this type of retrovirus, I will call it disarmed retrovirus. What will I call it? Disarmed retrovirus. So it is not going to cause any trouble. I am going to put my gene of interest and I am going to send it inside my body. This is disarmed retrovirus. So this is the commonly used vector in the case of animals. So if you are talking about animals, just remember, if this option was not there, something else was there and retrovirus is there, go for retrovirus, no problem. It's the same thing. Okay, but better, since it is being mentioned, you have to go for disarmed retrovirus. Cool. Chalo, next question. Enzymes which remove nucleotides from the ends of the DNA are. So there are two types of enzymes. Okay, so one, basically, you know, we have to cut the DNA, right? Suppose this is a DNA. Some enzymes cut the DNA from the ends. What do you call them? You call them exonucleases. Some of the DNA, they cut the DNA, some of the enzymes, they cut the DNA basically from the middle, and this is known as endo. They are entering and they are cutting, so they are known as endonucleases. So you have endonucleases, you have exonucleases, you have DNA ligase, you have DNA polymerase. So which enzymes would remove nucleotides from the ends of the DNA? From the ends of the DNA, you would be having what, children? Exonucleases. DNA ligase is for ligating two. Okay, I want to join two strands of DNA. For that, I would be using DNA ligase. DNA polymerase is for making a new copy of DNA. Okay, I hope you got it. Next question. Name the plasmid present in Agrobacterium tumefaciens. So this example that I gave, okay, suppose this was a bacteria whose name is what? Agrobacterium tumefaciens, and I told you it has a separate type of plasmid. Okay, what do you call it? You call it as TI plasmid. Why do you call it TI plasmid? TI stands for tumor inducing plasmid. And why do we call it tumor inducing plasmid? Because it has a specific gene over here, specific DNA which you call as tDNA. So TI plasmid has a DNA which is known as tDNA. You call it transfer DNA. So when this guy goes and infects the plant, what it does is that it transfers this particular tDNA sequence and it causes the whole trouble. This tDNA, when it is transferred into the plant cell, what happens? The plant cell becomes starts forming a tumor, and then they start releasing certain chemicals. So that is a problem. That is how it infects. Now what are we going to do? We are going to remove the tDNA portion and we are going to insert some of our gene of interest and then allow it to what say basically go and infect, because it's not a problem, it's anyway disarmed. I am putting my gene of interest there, but this is how it works, and the name of the plasmid is TI plasmid, tumor inducing plasmid, because it has a strand of DNA, a segment of DNA just known as tDNA, transfer DNA. Okay, so name of the plasmid present in agrobacterian tumor fissions is what, children? It is TI plasmid, tumor inducing plasmid. I'll write it over here also. Tumor inducing plasmid. Why? Because it has tDNA. And children, I want you to answer before I answer the questions. Okay, I forgot to tell about that. Yes, so the next question, you can try to answer this and then probably start off the video and check for my answers. So name the stain used to visualize DNA fragments in gel electrophoresis. So in gel electrophoresis, what is happening, children? Gel electrophoresis has a chamber like this where there are certain wells. Okay, wells as in depth there are certain areas. Okay, and there is electrodes being attached over here. There is one electrode over here, there is one electrode over here. This is known as cathode, which will be negative. Okay, and this will be anode, which will be positive. What happens? I put my DNA into these wells, and this particular thing is made up of agarose gel. Agarose gel, okay, which we take from an organism. Can you name the organism's name? It will be really good. I want to check if you know that as well. So what happens is that we cut the DNA and we have to, we are basically separating them on the basis of their DNA length. Okay, on the basis of their length, we are separating them. So the ones that would be having bigger length would be staying at the back position. The one that will be having smaller length would be coming over here. They would be traveling faster. So they would be reaching first, and over here you would be able to see the clear segregation. So these will be the ones that would be of basically they are larger DNA strands, then these will be the ones that are smaller DNA strands. So DNA is negatively charged, which is why it moves away from the cathode and comes to what? The anode. But again, I cannot see the DNA which is being put over here. So what do? I take a nitrocellulose sheet. Nitrocellulose sheet. And then I put it over here, and then I have it over here. Okay, this is the sheet. I have my DNA now transferred over here. It is still not visible. What do? I treat it with a stain which is known as ethidium bromide. This stain basically goes and attaches over there, but still it gets into the DNA. Okay, but still I would not be able to see until and unless I have put UV rays on it. Until and unless I put UV rays over it, I would not be able to see. When I put UV rays, it will appear to me in bright orangish color. Cool. So your answer would be what? Ethidium bromide. My dear children, I hope I am explaining the answers well to you. You're understanding the content, right? Chalo. Next. The enzyme responsible for the breakdown of X-gal. Okay, X-gal is a content. Okay, I'll tell you. X-gal is a content. Remember it. Okay, is a protein or whatever thing is there. Now, the enzyme responsible for the breakdown of X-gal is beta-galactosidase. What is the enzyme's name? Beta-galactosidase. This is going to break down this thing. The cells containing recombinant plasmid will produce C because D. What? I am not able to understand. So basically, recombinant plasmid means the one that is having my gene of interest. I have removed some segment from that plasmid and I have put my gene of interest over there. So the thing that happens over here is that in this particular plasmid, usually there is a segment. This segment is there for coding a gene that would be producing an enzyme that is beta-galactosidase. Okay, that will be what? Beta-galactosidase. Now, whenever X-gal is put in this particular medium where this particular plasmid is there, what will happen? This beta-galactosidase will be acting on X-gal and hence it will be giving out blue colonies, blue coloration, which is why we say it's a blue colony which is being formed. This is a normal case. But now, what I am going to do is, I am going to remove this area which is going to code for beta-galactosidase, and instead of that, I am going to put my gene of interest over here. Okay, so that means I am basically making it inactivated. I am basically making it inactivated. Okay, just give me a second. So over here, what happens is that now this particular colony would not be showing the color that is blue, hence it will be white, hence it would be white. And why is this so? Because beta galactosidase, it is what? Inactive. So your answers becomes what? You have to see for white colonies, and then you have to show that beta galactosidase is basically becoming inactive, which is the C option. So this becomes the answer. Yes. So next question. I hope you understood that question. Let's go with the next question. Choose the correct sequence of gel electrophoresis. Okay, gel electrophoresis. Now let's see. Gel electrophoresis is the thing that we already did, okay, which was mentioned like this. So the cutting of DNA by restriction enzymes, staining by ethidium bromide, exposure of UV rays, separation of DNA fragments under the influence of electric field in agarose gel. First thing, we have to cut the DNA, right? After cutting the DNA, basically we are going to separate it. So first step would be this one. The cutting of DNA by the restriction enzymes. After we have cut, now we are going to separate them along with their fragment length. So separation of DNA fragments under the influence of electric field in the agarose gel, this becomes the next point. Okay, so this is the first point, this is the next point. Now after that, we have to look for it, we have to see the fragments, right? For that we have to stain it with ethidium bromide, and still it won't be visible. For that we need UV rays, so this will be the next step. Okay, so we have 1, 2, 3 and 4. So 1, and then you have 4, and then you have 2, and then you have 3. This becomes your answer. Cool. Okay, so over here you can eliminate these options at the first chance only, because this is 1 and 1. So you know anyways this is going to be the first one. Okay, apart from that there is no other chance for things to be becoming first. And then you have to separate it, so this becomes the next one. So this is the correct answer. Going ahead. The host cells which have the ability to incorporate foreign DNA within them are known as. So the host cell, for example, this is a bacteria, this is the host cell, and I am saying that it has the ability to incorporate my gene of interest, or basically the DNA. What do I call this host cell? It has the ability, it has not gained until now, but it has the ability. So what do I call it? I call it a competent host. I call it a competent host, my dear children, because it might have ports, I might have made it undergo heat shock treatment and stuff like that. Now it is able to take in the DNA, or basically gene of interest. So the host cells which have the ability to incorporate foreign DNA within them are known as competent host. Cool? Works? Next thing. Autonomously replicating a circular extrachromosomal DNA of bacteria is known as. Autonomously replicating. What do you mean by autonomously replicating? As in, it does not need any force, it is just replicating itself. Okay, but okay, over here this is extrachromosomal. So I told you, if this is the bacteria, it will have its own DNA for sure, which will be coding for proteins. Apart from that, it also has certain circular things like this, which you call as extrachromosomal. This is extra from this particular chromosomal thing. So this is extrachromosomal DNA, which is not going to code for any protein or stuff like that, but it can replicate. It can definitely replicate, my dear children, and we are going to use this thing for acting like a vector. So this is over here, I make a cut over here and put my gene of interest. So this thing that you see over here, what do you call it? You call this as plasmid. It will have its own origin of replication and everything, just like this guy, but it does not code for any protein. So even if I make any changes over here, it's just not going to make any much changes in the bacteria. Okay, usually. Now there are different type of plasmid. For example, TI plasmid. If I make a change over there, that bacteria would not be able to basically infect, but it will still survive because it has all the other proteins coming on from its original chromosomal DNA. Okay, so that is separate thing. So autonomously replicating circular extrachromosomal DNA of bacteria is known as plasmid. I hope you understood that as well, and with this we are going to complete all the 1 marker question. Now these 2 questions came in your model question paper as a fill in the blanks question, so please understand that. Now let's get into the 5 marker questions, and the first one itself is very much interesting. Given below the diagrams of plasmid A and B, observe meticulously and answer the following questions. So over here I want you to have a look at this picture first. So these are the 2 plasmids. Okay, this is a gene that is going to code for ampicillin resistance. Do you understand what you mean by ampicillin resistance? So that means this gene is going to code for something which would make the bacteria resistant to something that is known as ampicillin, a antibiotic. So even if I am putting antibiotic to kill this particular organism, which antibiotic? Ampicillin, it won't die because it has an ampicillin resistant gene. Similarly, there is another one that is known as tetracycline. TET stands for tetracycline resistant gene. Over here, this is a plasmid, and now you have different plasmid which has LAG-Z gene. Now LAG-Z gene is the one that codes for beta galactosidase. Remember the X-gal experiment, the blue colony and white colony one. So LAG-Z gene is going to code for beta galactosidase. Okay, now listen to me carefully. Okay, I'm just going to write. So over here you have, that means it is ampicillin resistant. TETR, that means it is tetracycline resistant. And then you have LAG-Z, that means it is producing beta galactosidase, and this beta galactosidase is going to convert X-gal to blue colored compound. Okay, it will, when it breaks down X-gal, it will give blue colored thing. Now, which plasmid is or are you select for cloning and why? Which plasmid will you select for cloning? I would say I can select both this as well as this. Why this? Because it is kind of easy. It is kind of easy to identify plasmid B. Okay, so why? In them, I am just making sure that basically this is the plasmid. If I am putting my gene of interest over here in the place of LAG-Z gene, I would not be having beta galactosidase, and hence even if I put X-gal, the colony would not have any color change. It would be white itself. It would be white itself. That means this particular colony has what? My recombinant DNA. Recombinant DNA. If in the colony I had certain areas where blue color is present, that means that is not recombinant. So it is very much easy for me to understand that, hey, recombination has definitely taken place over here. So I'll definitely go for plasmid B in first course. Second thing, I can also take this plasmid Y. Basically, what can I do is, I can take one gene, that is for example tetracycline. I can put my gene of interest over here. I can put my gene of interest over here. Okay, when I put my gene of interest, will it show tetracycline resistance? No. But will it show ampicillin resistance? Yes, because that gene is still there. So how will I be able to understand which one is recombinant, which one is not? The one that would not be showing tetracycline resistance would be the one, okay, the one that shows ampicillin resistance but not tetracycline resistance will be the one that would be what? Recombinant. That would be the ones that are recombinant. So one that does not show the resistance for tetracycline because it is insertional inactivation. I have inserted my gene of interest and hence it is inactivated, but it is still showing ampicillin resistant gene. That means this is a recombinant DNA. Suppose it is showing both resistance, that means it is not recombinant. That means the DNA has not been come up over here. You get it? So this is how I can use both of them, but again this is less time consuming, that is plasmid B. So I can take both, this would be the answer. Okay, and there is a term, what is insertional inactivation? I think I mentioned over here also. Insertional inactivation means when I am putting my gene of interest into the space where another gene was there, and hence because I am putting it over here, this particular gene which was usually there, it got inactivated. Now it is not able to produce beta galactosidase. This is known as insertional inactivation. Over here, when I am putting my gene of interest over here, what happens? It is now not tetracycline resistant. This is insertionally inactivated. That's the meaning. So how will you write the answer? Both plasmids allow for cloning via insertional inactivation, but plasmid B might be preferred for ease of screening using the blue-white method. Blue-white method as in the blue-white colony method. You can write the answer like this. If you want, you can explain both. That basically I can use both, but this will be less time consuming because it will be easily giving out white colonies and I would be able to identify which one is recombinant, and if it is blue I would be understanding it is not recombinant. Similarly over here, if it is ampicillin resistant but it is not tetracycline resistant, I can identify it is recombinant. If it is resistant to both of them, okay, that means it is not the recombinant, but it is again a time taking process because you might have to include different plating, okay, different cultures. Now what is insertional inactivation? It is a process where insertion of foreign DNA into a marker gene. Okay, these genes I call it marker genes. Okay, for example, tetracycline in plasmid A or LAGZ gene in plasmid B disrupts its normal function. This disruption results in detectable phenotype such as what? Loss of tetracycline resistance in plasmid A, loss of beta carotenoid activity in plasmid B. Phenotype as in the physical appearance. So basically it is going to bring out certain changes. As of now, it is not going to produce beta carotenoid or produce basically resistance to tetracycline. So that is a change that occurs. So you can simply write whenever the gene of interest is put into the area where other gene was present, and this gradually makes the other gene not be able to produce its product. This is known as insertional inactivation, or when that particular gene becomes inactivated because of insertion of the gene of interest, it is known as insertional inactivation. Now, next question, will the number of culture plating you should make is same or different to select a recombinant if insertional inactivation is possible? They are asking you simple terms. In simple terms I will tell you, okay. They are asking you, will the number of culture plating, that means how many times you have to culture and everything, it would be same or different if you have to select the recombinant. What is the case in both of them? So basically for plasmid A requires two-step plating. There will be two-step plating. First, plate on ampicillin containing media to ensure plasmid has uptaken replica. Wait. I'll explain this to you, okay, otherwise it will be confusing for you. So this is, okay, this is one guy. Okay, the first thing that I will do is, I have my plasmids, okay, I have my plasmid. Now over here, I am putting my gene of interest at this particular location. Agreed? Okay, in the tetracycline resistant gene. Now, there can be two scenarios. This plasmid might have gotten into the host. This is a host cell, this is the next host cell. So it might have got into the host, it might not have got into the host. This one I would call it transformed because there is a plasmid inside. This one I will call it non-transformed, or basically non-transformant. Why? Because it does not have the plasmid. But how will I get to know that? How will I get to know whether the plasmid is there or not? Okay, I have to make sure of that also. So for that, what will I do? First thing, I will be treating it with, can I treat with tetracycline? I cannot treat it with tetracycline because anyways tetracycline is not there over here as well. So both of them will be negative. So I will treat it with ampicillin. So I will get to know that this is resistant, this will survive, this is not. If this is not, that means this does not have the plasmid in itself. This does not have the plasmid only. I am not talking about recombinant DNA, nothing like that. Whether it has a plasmid or not, it does not have the plasmid only. So first step is basically I have to plate to ampicillin resistant containing media to ensure plasmid uptake. Now I am sure the plasmid has been taken up. Second thing, I will make a replica plate of it, and then what say I will plate on tetracycline containing medium to identify the recombinants. Now what will I do? Basically I will put tetracycline, I will create a replica plating where this time I am going to treat it with tetracycline, and then I will come to know if it is able to resist, that means this particular thing is not a recombinant. This particular thing is not a recombinant. So for example, you think like this. I have the plasmids like this also. I have the plasmids like this also. It can also be taken up. So this is the next scenario, okay. So I hope you got to understand about transformants. Okay, this is the non-transformant, this is the transformant. How did I come to know that? With the help of ampicillin. Now there is one more scenario that can be possible, and that scenario is what, children? Over here, just imagine that the plasmid that it got was like this. So this would also be ampicillin resistant also. This will also show this is also transformant now. So you have two transformants now. But how will you come to know which one is the recombinant one? So transformants and recombinants are different. Transformants is the one that has uptaken the plasmid. Now you have to make sure whether it has uptaken a plasmid that has my gene of interest, or whether it has uptaken a plasmid that does not have the gene of interest. For that, how will you understand? Basically, I will try to treat it with tetracycline. This is second plate. This is replica plate where I will be treating it with tetracycline. I will get to know that this guy which is there, it won't be able to survive. Why? Because it does not have the gene. Whereas this one is able to survive. That means it is not recombinant. So this is something that I have to exclude. This is what? A non-recombinant. But this is what? A recombinant. So the first plating was done with the help of ampicillin. I got to know which one of them has the plasmid. Okay, second thing, I did it with tetracycline. I got to know which one was able to survive. If it was able to survive, that means it is not recombinant. It has the gene for tetracycline. Over here, this gene is inactivated because I have inserted my gene of interest. Get it? So I need two-step plating. But for plasmid B, where there is lag Z gene, over here I just need one plating. So contains the lag Z gene, which allows for insertional inactivation using the blue-white screening. Recombinant disrupt the lag Z gene, leading to white colonies on X-gal media. Non-recombinants produce beta-galactosidase turning blue on the same medium. So in the same medium only these things are happening. Over here in one particular plate only these things are happening. The one plasmids, okay, the colony, the plasmids that would be having the lag Z gene, it will be blue in color. The one that does not have would be what, children? White in color. So which one is recombinant? The white one is basically recombinant. Why white one is recombinant, or why blue color is not the recombinant? Because blue color is shown when beta-galactosidase activity is there. Okay, so that's why. So over here it's not there, which is why it is a recombinant one. So this is how you have to answer it. I know it's a bit tricky question, but with this you get 5 marks. If you understand things, write in your own words. They won't judge you for the complicated terms that you are using. It's okay. This chapter itself is complicated in a bit way. Okay, so you have the whole right to make it complicated and right, but underline the things that you are trying to show them. Okay, it becomes more visible. Okay, so that is one thing. Now, study the diagram given below and answer the questions that follows. What is Eco R1? So Eco R1, what do you think is Eco R1, children? Eco R1 is an enzyme. What enzyme, children? Restriction enzyme. It is what? A restriction enzyme. Okay, and now where do you get it from? You get it from Escherichia coli. So over here E is this, CO is this. Now R1 is the basically sample number. Over here you have RY13. From there you got R1. Okay, so this is a strain number, not sample number, strain number. So Eco R1 is a restriction enzyme, type 2 restriction endonuclease that recognizes specific DNA sequence, that is 5-GAA-TTC. Please remember it, 5-GAA-TTC, and it cleaves between what? G and A. It is going to make a cut between G and A. And it is derived from Escherichia coli strain RY13, strain RY13. So you took E from this, you took CO from this, you took R from this, and this is R1. So in the order of how the strains were discovered, you give it name. So this was discovered first, so you got it Eco R1. Okay, in their category it was discovered first. Now this is a restriction endonuclease. I told you it's an enzyme. You have exonuclease which cuts at the end, and endonuclease which cuts from the middle. This is an endonuclease. Okay, and the sequence is this. Now what is the next question? What is the action of, how is the action of exonuclease different from that of endonuclease? I already told you, exonuclease basically cut from the end, endonucleases would be cutting from the middle, my dear children. So exonuclease removes nucleotides from the end of the DNA or RNA strand, either 5' or 3' end. So loss of tetracycline resistance, okay, now next thing, endonuclease cuts within the DNA or RNA strand at specific sites, breaking internal phosphodiester bonds. So they are going to break down some bonds which are known as phosphodiester bonds. So between our nucleotides, which forms the DNA, there is bonds which are known as phosphodiester bond, which is known as what? Phosphodiester bond. So this is being broken down there. Now Eco R1, okay, next question. How are sticky ends formed on a DNA strand? Why are they called so? So what are sticky ends, my dear children? I told you, basically, suppose this is a strand, okay, and over here you have, this is the 5' end, this is the 3' end, you have GAA-TTC. Similarly over here also you would be having GAA-TTC. This is 3', this is 5'. Now your restriction endonuclease, that is Eco R1, will make a cut over here between G and A. Similarly, it will make a cut over here. Okay. Now what do you get after making it cut? It will look something like this. It will look something like this. This is one strand. Okay, now you have to make the other strand also. So other strand looks like this. So over here, if you see, this is going to act like sticky end. Okay, this is going to act like what, children? Sticky end. Why it is going to act like sticky end? So if I make a smaller picture, I think it will be more clear for you to understand, rather than having a bigger picture like this. Okay, if I just focus on this, I get something like this. This is 3 dash. Okay, and over here I have C T T A A. This is one particular strand. So over here there is no one to form complementary strand. Over here there is no one to basically form a complementary strand. Why? Why there is no one to form complementary strand? Because I have separated from here, and it is also going to make another strand like this. Another strand like this, which is having AATTC. So this is going to act like a sticky end. Okay, this is going to act like a sticky end. Similarly, this one is also going to act like a sticky end because there is no complementary pair. I have cutted it, I have segregated them, separated them. These ends are free. Now this can go and bind to something else. So these ends that you see over here, they are known as sticky ends, and they would be able to make complementary base pairs with anything that has this pairs. Okay, that with any other strand which has this particular pairs. This is very much important. So I make a cut over here in my gene, in my DNA, and with the same enzyme I am going to make a cut over here in the plasmid DNA. What happens? They will be having similar type of sticky ends. So this is one sticky end, this will be one sticky end. I put my gene of interest. Over here. So this sticky end matched with mine, and the sticky end over here matched again with mine. So over here, with the help of the same restriction enzyme, if I cut, I get similar type of sticky ends, and they will be able to make complementary base pairs again. So this thing that I am doing is the formation of sticky ends, which is very much important in the process of recombinant DNA technology. So over here also you can see it clearly. Why didn't I make it? I don't know. So I made a cut over here. You see, what do I get? This is the end, this is another end. Okay, now this area is free, this area is also free. If I have something else, I have recombinant DNA, it comes and attaches together, and now it's all cool. So how do you answer this? EcoR1 cuts DNA at its recognition sequence, that is this, in a staggered manner, leaving overhanging single stranded ends. So I make a cut over here, I make a cut over here, and I make a cut over here. These overhangs can easily form hydrogen bonds with complementary sequences, allowing them to stick to other DNA fragments with matching ends. This property is essential for ligating DNA fragments. Ligating as in making the DNA fragments come together. Okay, during the recombinant DNA technology. So I have to make sure that I am using the same restriction enzymes both in my host and also the place where I am getting my gene of interest from. So how are sticky ends formed? Do you know that? Basically when they cut, they are going to make a staggered cut there. And why are they so called? Because they can easily form hydrogen bonds with the complementary base pairs. So that is done. Next question. Write 3 vector free techniques that can be utilized to transfer recombinant DNA in a ready host cell, that is competent host cell. So over here I want to put my gene of interest, basically my recombinant DNA. I am not going to use a vector, so I cannot use bacteria and things like that, like agrobacterium tumescens and stuff like that. So what are the other ways that I can use? I have already told you about gene gun, where I am going to take microparticles of gold and tungsten, and I am going to coat it with DNA, and I am going to shoot it with high velocity inside the nucleus of the plant cell. That is one way. So what do we have? We have gene gun. Okay, apart from that, what do we have? We have micro injection. This is done in plants. We have micro injection, my dear children. Micro injection, where basically with the help of a needly thing in the animal cells, I am going to insert the DNA directly into the nucleus. This is done in animal cells. Next thing, in bacteria and all, I can take heat shock treatment. What do you mean by heat shock treatment? Basically, I am going to put the bacteria under very high temperature and then cold, then I heat, then cold. So basically what happens, there will be pores that will be made in their membrane, and with the help of the pores which is there being made in the membrane, I also use certain ions also for creating the pores and stuff like that. With the help of these pores, I am going to transmit my gene of interest inside the host. So this is another way. So over here, these are the ways you can write it properly. Electroporation, same thing that I said. Over here, high voltage electric pulses create temporary pores in the host membrane, allowing recombinant DNA to enter. So this is different from heat shock. Okay, this is another method, that is electroporation, where I am using what? Electric pulse. Okay, when I am using current, I am using current to basically create what? Pores in the host cell, so that things can easily enter. Apart from this, I can use heat shock treatment as well. Then we have microinjection, where DNA is directly injected into the nucleus of the host cell using a fine glass micropipette. This is done in animals. Gene gun, that is also known as biolistics, where a DNA coated microscopic particles like gold or tungsten are shot into the host cell using the gene gun. Now this is done in the case of plants, and this is done in the case of bacteria. Okay, done. So that is all. Now agrobacterium tumefaciens acts as a natural vector for cloning genes and plants. Substantiate, I already told you, agrobacterium tumefaciens has what, children? TI plasmid. TI plasmid has what DNA? tDNA or transfer DNA. With the help of it, goes and infects the plant. Now if you remove the tDNA, if we put the gene of interest, it becomes our vector, right? So this is the same thing. Mechanism, Agrobacterium tumefaciens contains TI plasmid, okay, that can transfer a segment of its DNA, that's tDNA, into the plant cell using infection, during the infection, right? This tDNA integrates into the plant genome. Whatever tDNA it is there, it is going to integrate into the plant genome. This is the usual stuff. Now how do we use it? Scientists modify the tDNA region to carry desired genes instead of what? Tumor causing genes. The bacterium then serves as a natural delivery system to insert basically what? Recombinant DNA into the plant cell. So there it was putting its own tDNA, now it is going to put our gene of interest over there. That's how it can be utilized. It's a funny question, no? Not funny question, fun question. So yes, that is there. My head is aching, I don't know why. Okay, cool. Now, next question. Why there are 17 questions? This is also 17, this is also 17. So this has to be 18. Okay. Write any 4. This is 18. Write any 4 tools used in recombinant DNA technology. Okay. Write any 4 tools that is used in recombinant DNA technology. Mention any 2 methods of introducing alien DNA into the host cell. I already told you, biolistics, anything you can say. Electroporation or basically gene gun method, microinjection, everything. Okay. Name the stain used to visualize DNA fragments in gel electrophoresis. You already know it. What is it, children? Ethidium bromide. So write any 4 tools used in recombinant DNA technology. This is something that is new to us. Let's see that. So the 4 tools are enzymes. You need enzymes, restriction enzymes. You need vectors that could carry. You need host organism which could carry the gene of interest now. Okay, you have to transfer it to some organism, no? That will be host organism. You need bioreactors. What are bioreactors? Bioreactors are something where the bacteria and everything could basically amplify in number. They could increase in number, and there we get the the product that we are looking forward for. So these are the 4 tools that we need for recombinant technology. Enzymes, vectors who can carry, host who can take this particular recombinant DNA and basically make it multiply in their body, and then bioreactors. Now methods, microinjection, biolistics, vector mediated, heat shock, disarmed pathogen vector, anything. Now you have ethidium bromide, that is the stain that is used to visualize. Now next thing. Explain the process of denaturation, annealing and extension with respect to polymerase chain reaction. I already explained it to you. Okay, in the front line, define plasmid. What is plasmid? It is the extra chromosomal DNA, okay, which is there in the bacteria. Now it can be used in recombinant DNA technology. Why are restriction enzymes called molecular scissors? Restriction enzymes are known as molecular scissors because they are going to make cut in the DNA. Okay, and DNA is a molecular thing, right? So this answer is, they are called molecular scissors, then they are used to cut the DNA precisely. Plasmid is what? Is an extra nuclear or extra chromosomal self-replicating. Yeah, please mention self-replicating double-stranded circular DNA of bacteria. It is double-stranded, that is why it is made like this. Okay, and what are the three very important steps of PCR, my dear children? Denaturation. First, the DNA strand is there, you have to open it up, right? This occurs in 95 degrees Celsius, 92 to 95 degrees Celsius. So here it is 94, so you can go with that. At this particular point of time, the DNA will get segregated, okay, separated basically. Then annealing of the primers. Okay, annealing of the primers means the primers would come and attach on the strands, okay, on the 5' end, so that the next step can happen, that is DNA elongation can happen. So in annealing, the temperature is around 55 degrees Celsius, that is 50 degrees Celsius also it works. So two primers paired with complementary base sequences present in the ends of the separated DNA strands. So at the ends of the separated DNA strands, the primer will be coming and attached. What side? It should be, this would be in 3', this in 3', this is 5', 3'. So over here it comes and attaches at this particular end, but opposite it should be 3', and here this particular end that you see where the primer is going to attach is 5'. If I am making you confused, I do not want to do so. So suppose this is a DNA strand, this is 3' end, this is 5' end, this is 5' end, this is 3' end. Your primers would be coming and attaching over here, such that the next step, DNA polymerase can extend in 5' to 3' direction. Cool? Okay, now extension of primers. The temperature of the reaction mixture is increased to 72 degrees Celsius. Okay, this temperature is suitable for the activity of DNA polymerase. What do you call it? Taq polymerase. Apart from DNA polymerase, what do you need? You need nucleotides also. So what do you call these nucleotides? You call this as deoxynucleotides. Okay, the one that makes the DNA, the segments, the small segments, you call them deoxynucleotides. Now deoxynucleotides would come together to make the DNA, extended DNA. So that is there. Cool. Now we have, you can go and read this, okay, you will get the notes, please not worried, do not get worried, there is a form, you have to fill that form and then you can get the notes. Now draw a neat label diagram of simple stirred tank bioreactor. So you have to make things like this. How can we make? Let's make it together. So over here we have to make a small cylinder like this. Cool. Do it like this, and over here also. Let's make it like this. It's a cylinder, right? Now what do? That should be stirred, it is a stirred tank bioreactor, right? So it should be looking like this. And over here you should be having certain plates like this. So you make certain plates. It should not be difficult. Ayo. Please don't think the questions would not be coming like that, okay? Questions have come before. And I have seen this question so many times, which is why I put it. So you please do not think it won't come. So this is stirred tank bioreactor. Okay. I hope the teachers, everyone know that we are not basically professionals making over here, so you don't mind. Now, what do we need? We need a motor for this to run. This is the motor that we need for it to run. And then also we need a timer, so that we get to know for how long it is run. So this is motor, my dear children. Important things you can mark, other things if you're not needed, do not mark, okay? And now you have what? You need acid pH control over here. So for that, what do we have? We have something like this, which would help us for understanding the pH control. Okay, cool. Now over here you should be having a vent for the sterile air. To go through, okay? Then what? Over here you will be putting the broth, culture broth, where things and everything will be there. Okay? This is what the impeller, which will be moving around the blades, that means it's a blade. And sometimes it also have some temperature regulation coat also. At times it does have what? Temperature regulation coats as well, which regulates the temperature. So this is something that is important, nothing much. Okay, at least this if you draw, you'll get good things. Okay. Now there are so many vents, my dear children. Actually there are so many vents opening up here, taking up here. So I'm not making all of that, which is not there in your NCERT. So let's be restricted to this only. Okay. Now next thing. Draw a neat labeled diagram of PBr 322. This is very much important. PBr 322. Okay. For that you have to make 2 rounds because it's a plasmid. Agreed? It will have its own origin of replication. So I'll say ORI site. Now, apart from origin of replication is for DNA polymerase to initiate the process of replication. Apart from that, there are certain proteins also which are being required for replication. You call it ROP. Okay, what do you call it? ROP. Now, you have very important sites. One is ampicillin resistant site and the other one is tetracycline resistant site. In the ampicillin resistant site, or basically let's start from tetracycline. Over here you have BAM-H1. Everyone is 1 for some time, okay? Then you have SAL1. SAL1, you can remember that, okay? BAM-H1 and BAM-H1 and then SAL1. These are what, children? These are restriction enzymes sites. So here BAM-H1 can come and cleave, here SAL1 can come and cleave, but this is gene for tetracycline resistance. Similarly, ampicillin resistant gene, also over here also you find certain things. One is what? PVU1 and the other one is what? PST1. PVU1, PST1. Now there is something that is PVU2, that is another enzyme which makes a restriction site over here. So this is PVU2. Okay, this is what? PVU2. Now you have 3 more enzymes. Okay, one is your favorite EcoR1, then you have clavin, then you have HIND3. So one is EcoR1, then you have clavin, then you have HIND3. This is what your PBR 332 looks like. Okay, so there is ampicillin resistant gene. This whole thing that you see over here, this is ampicillin resistant gene. This is tetracycline resistant gene. Within tetracycline resistant gene, you have 2 recognition sites. One is for BAM-H1, other is for SAL1. Similarly, over here you have PVU1, you have PST1. PVU1 is there, so PVU2 is there over here in ROP site. Okay. Now over here you have 3 enzymes. One is EcoR1, then you have clavin, then you have HIND3. This is the site for what? Basically PBR 322. You have to remember it. There is no other way. We cannot skip it. And with this, for the first time, we have completed the question in 1 hour. And with that, I want to tell you one thing. I hope this particular video was useful. Okay, I looked for the questions. The questions were like this only. It was so repeated. Okay, so you can expect these type of questions coming off from this chapter, nothing else. So you can have a thorough read of the NCERT also. Once you're done with the video that I have mentioned over here, go read your NCERT and it will tickle up your brain. You will understand things lot better. And now there is a PU2 success blueprint test series as well. If you are someone who is interested in that, definitely try to avail it. The video link is given in the description box. It is of 599 rupees, where we are going to provide you 12 mock tests. My dear children, there will be 4 part tests. Part test means basically in one particular question paper there will be different subjects coming on together, and there will be full length test as well. 8 of them will be full length. That means individual test will be there for subjects. Now, apart from test series, there will be also video solutions, and apart from video solution, there will be doubt clarification classes as well. So, if you are someone who is interested, do not think twice, go check it out. And if you have certain doubts in whatever I have taught today, definitely do not forget to comment it down. I would rectify your doubt. If you love my class, definitely show your love, and that's all. So, I've got a bit of headache and things like that, so I would just stop the video, and lots of love to you guys. Not be panicked, everything is going to be really good. Just take care of yourself and have that sweet smile of yours. So bye bye, take care.